Michelle Staros and Hannah Erdvig would like to make an announcement about their TA-ship and work in here.
I'm Michelle Staros. I am one of the co-instructors for the lecture section. I'll be teaching Unit 4. I'm also the course coordinator for recitation. But previous to this, I was actually a GTA, and previous to that, a UTA. So I have kind of been through all the ranks. I'm very approachable, I promise. I am the queen of mispronunciation. I am a massive klutz, and I do stupid things on a regular basis. My latest was when I went shopping after work, and I did not want to have to bring my laptop in my purse, A, because it's heavy, and B, because I'm always terrified they're going to think I tried to steal it, even though they don't even sell that laptop. So I shoved it under the car seat, and then the next day, couldn't find it. Until I finally remembered that I had left it in my car all night long, unlocked, in the freezing cold. Fortunately, it still works, thank God. But that's me, so come see me, please. I have office hours on Tuesdays from 12 to 2. And here's Hannah.
Thanks, Michelle. I'm Hannah. I'm a GTA this semester. A few random facts about myself. I have a dog named Turtle. I promise it makes sense. I also have been told by several people that there's this spot on the side of my nose that twitches when I think really hard. And I hope someday I'll learn enough about the human body to make sense of that. I have office hours today, 4 p.m. to 6 p.m. and every Friday. You can follow a link on Canvas, and I'm really excited to meet you all.
Thank you.
Yeah, again, use these TAs. I mean, they're here to help, and often they'll sit alone in office hours and there's no one there. And they're very helpful, particularly if you're one of the students who's taking this course for the second or more time. Please spend some time with these guys. They can help you through this course, because I know that the thinking in this course is a little different than many other courses. The questions are different on the exams, and they can coach you through this. And I think if you do that, it's going to help you with all your classes. So what are we going to do today? Well, we're now going to move on to endocytosis. We'd started this in one of the two sections, I don't remember which was which. And endocytosis is going to be the other side of the coin from exocytosis. Yesterday's discussion was exocytosis and the idea of those three kinds of proteins that we generated. The first kind was the transmembrane proteins, the secreted proteins, and the third kind was the lysosomal proteins. And we'll begin today with removing those lysosomal proteins from the Golgi apparatus to the lysosome. And then we'll talk about how we can use those proteolytic enzymes, which are the lysosomal enzymes. How do we employ them in the lysosome? And the way we're going to talk about employing them in the lysosome is we're going to bring in potentially pathogenic bacteria from the outside world in a process called endocytosis. The story here is going to be about a particular cell type called the macrophage and how it can grab on to potentially pathogenic bacteria and bring them into the cell and deliver those bacteria to the lysosomes. I don't want to leave you with the idea that that's the only form of endocytosis. There are any number of different forms of endocytosis. When we get to the nervous system, if you take a course in neurobiology, you'll see that one of the places where you get endocytosis there is in recycling of vesicles. But our purpose here is simply to contrast the exocytotic event with the endocytotic event and then connect the endocytotic event with the lysosomal, that intracellular compartment that contains the witch's brew of enzymes. So we'll talk a little bit about vesicle trafficking from the plasma membrane to the lysosomes. Then we'll spend some time in mitochondria. Mitochondria is the last of the organelles we're going to talk about. Mitochondria are really, really important to us. They are the sites where we generate most of the ATP inside the cell. And they have a peculiar way of generating ATP. The ATP is generated by using oxygen as the final electron acceptor, and we pass the electrons down the electron transport chain. We're not going to get into that detail. That's a biochemistry course, perhaps a cell biology course. Here, all I want to emphasize is that we have this organelle, and it's kind of peculiar. And it's peculiar because it has a double membrane. And we'll see that it has its own set of genes. The genes are dedicated almost entirely to making ribosomes and transfer RNA. But it has its very own set of genes. It has its own set of ribosomes. And so from that, it appears that these mitochondria were originally proper bacteria. And they were captured maybe 1.5 billion to 2 billion years ago by some predatory, primitive And all of cells on the planet, whether they're plant cells or animal cells, are related to that cell which captured that mitochondria, that bacteria. And we'll talk a little bit about how those ideas were sorted out. And the fact that mitochondrial inheritance is unusual. Mitochondrial inheritance is all through the matrilineal line. So all of your mitochondria came from your mother, and all of hers came from her mother, all the way back to the beginning of time. And so we'll talk a little bit about that and what the implications of it. Then we'll move on to the cytoskeleton. You may remember that one of the major differences between most of the eukaryotes and the prokaryotes is the eukaryotes have a highly developed cytoskeleton. And we're going to talk about the cytoskeleton with three major players: microtubules, filamentous actin, and intermediate filament proteins. Most of our time is going to be spent on actin and microtubules. What all of these protein types have in common is that they themselves are protein polymers. In other words, they are polymers of individual proteins. We're going to take individual tubulin molecules and link them end to end and side to side and make these great long microtubules. We can do something similar with actin. We can take G-actin and glom it together one to the next to make filamentous actin. Now, if you go on to cell biology a little more, you'll find that the control of the formation of actin filaments is really more complicated than microtubules. There are a whole host of proteins that are regulating actin filament assembly. But for our purposes, we'll keep it pretty simple. And the idea we'll see is that we're going to make actin filaments as a polymer of actin monomer, and we make microtubules as a polymer of tubulin. The third player is going to be intermediate filament protein. And the intermediate filament protein are also polymers. And they're polymers of monomer intermediate filament protein. The two characters we're going to talk about here are keratin, which we're going to see a little later when we talk about keratinocytes in skin, and neurofilaments. Now, we're not going to spend a lot of time in here right now on these two because they don't support transport. And one of the main hallmarks, one of the main claims to fame for the microtubules and for the actin filaments is that they support the movement of vesicles. Vesicle trafficking can occur on those substrates. So we're going to spend most of our time concentrating on the assembly and function of filamentous actin and microtubules. One of the characteristics of these proteins, particularly the microtubules and the filaments, is that they have a plus and a minus end. I want to be a little careful in that terminology because when I say plus and minus, for some people that calls to mind the idea of charge, and electrical charge. It has nothing to do with electrical charge. It's much simpler than that. When we talk about plus and minus ends of these molecules, there's one end to which we add monomer and one end from which we take it off. It's not a stretch, therefore, to imagine that the plus end is where we add the monomer, and the minus end is where it comes off. So that's one of the characteristics of these molecules, is we can add them at one end and take them off at the other. And it's true both for microtubules and filaments. I'm going to make a microtubule, and we'll draw some microtubules on the board, and we'll talk about some of its functions. Once we talk about finally assembling microtubules and putting them together for transport, we're going to make them stable. I have to remind myself to put microtubule organizing center at the negative end. And we'll talk a little bit about that. But in both cases, the microtubule is going to support movement using a molecular motor. The particular molecular motor here is going to be kinesin. But the kinesin is said to be a plus end directed motor. It always walks toward the plus end of the microtubule. But there's another motor here involved with microtubules, that's called dynein. And dynein is a negative end directed motor. It marches in the opposite direction. And we'll talk a little bit about the implications of that inside cells. Filamentous actin also has an orientation, and it also has a motor. But in this case, the motor is going to be myosin. And there are going to be cellular myosins, which predominantly, but not entirely, are plus end directed, and we'll talk a little bit about that. And then when there's time, at the very end, we'll start talking about how we're going to use these motors as ATPases. One of the things I want you to really hold on to when we talk about the motors is that in all cases, they're enzymes that convert ATP to ADP. And in the process of converting ATP to ADP, they're converting chemical energy to mechanical energy. And we'll briefly talk about how that happens. But let's start here with our idea. Well, let me start here with a picture. I like this picture, and I didn't show this image before, and it really is meant to emphasize the structure of the two membranes that are forming the nuclear membrane and forming the endoplasmic reticulum membrane. And I like this picture because it gives me a sense of what it looks like to be inside the cell. All of this blue area in here, this blue C area, that's meant to be inside the nucleus. And this barbed wire kind of structure, that's meant to be the chromatin. Those are meant to be the chromosomes. But what I want you to look at is how there is a phospholipid bilayer right here. That's the nuclear membrane. But if we walk along this ridge, if we can imagine walking along this ridge as though it were a wall, we'll walk along and we'll come to this site. That's going to be a nuclear pore. You can't jump over the nuclear pore. But in fact, if we can walk on this, we'll have phospholipids. And the phospholipids character will change a little bit. They're specialized phospholipids right here at the sharp turn. But we convert now from being the phospholipid bilayer of the nuclear membrane to the phospholipid bilayer of the endoplasmic reticulum. And then these open pores, the nuclear pores, these are now continuous with cytoplasm. So we can now move molecules from the nucleus out into the cytoplasm and back in again through those nuclear pores. But the rest of the membrane, the nuclear membrane, is then extended into the endoplasmic reticulum. And each of these yellow buttons is meant to be a ribosome, and that constitutes then the rough endoplasmic reticulum. Now, this doesn't show how we're going to butt it off. That was yesterday's story, how we're going to butt off membranes and carry those proteins which are in the lumen, in the opening of that structure, to the next step, the Golgi apparatus. Yesterday's discussion was focused, concentrated on the faded vesicles, where if this is the cell, and this is the nucleus, we said that the traffic in the cell was from the endoplasmic reticulum, and I won't emphasize too much what we just put on the board, but this is now the ER. And the vesicular traffic was from the ER to the Golgi apparatus. And so these cargo-carrying containers would start out in the endoplasmic reticulum, go to the Golgi, and then move their way, moving from one part of the Golgi to the next where we completely finish those proteins. And then from this end, from the trans end, where we finished is we would generate a vesicle. The vesicle would become free of the Golgi apparatus, find its way to the membrane, and then it could fuse the plasma membrane in a process known as exocytosis. We finished with the idea that there were another set of proteins that could be generated in the ER. These are the lysosomal proteins. And these vesicles coming off the Golgi apparatus were destined to go to this structure here called a lysosome. And when the lysosomal vesicle fused with the lysosome, when this vesicle carrying lysosomal proteins left the Golgi apparatus and was moved to the lysosome, it fused with the lysosome to deliver those lysosomal enzymes. That generated this membrane-bound compartment where we had a whole series of proteolytic enzymes that are dedicated to destroying proteins. Some of these proteins will also be dedicated to destroying lipids. What I want to do now is I want to change the story a little bit and imagine that this isn't just a garden variety cell. This is a specialized cell. This is a specialized cell called a macrophage. Macro meaning large, phage meaning eater. These are the big eaters. These are endocytic cells. What that means is that these cells have the capacity to bind on their surface, potentially a bacterium. This bacterium may be pathogenic. It could be harmful to us. And so there are proteins that allow the bacterium to stick to the macrophage. The sticking of the bacterium to these proteins starts a sequence of events. Starts a sequence of events such that this portion of the plasma membrane begins to deform, begins to change shape. And as it changes shape, it invaginates. And as it invaginates, it's carrying with it the bacterium that's stuck to those proteins. This process is known as endocytosis. Exocytosis was out of the cell, endocytosis is into the cell. And what will happen is as we invaginate that membrane, eventually we reach a point where the vesicle pinches off. But now that vesicle is carrying in its lumen the bacterium. And this vesicle carrying the bacterium, this vesicle with its encased bacterium, can now be targeted, it can be moved to the lysosome. And this vat of proteolytic enzymes is here waiting for it. And when the vesicle fuses with the lysosome, it delivers the bacterium into the lumen of the lysosome, and now this bacterium can be destroyed by the proteolytic enzymes. This is the process. Now, I don't want to leave you with the idea that this is the only function or role of endocytosis. Endocytosis is occurring in all cells all the time. It plays a really important role in getting things from the outside world to the inside. And not everything coming from the outside is destined to go to the lysosome. But I like this story because it gives you an idea of the function of the macrophage. It gives you an idea of how we can direct vesicles to a particular site inside the cell of the lysosome. But not all endocytic vesicles are destined to go to lysosomes. If there's time, we talk about the nervous system. One of the things that happens in the nervous system is after vesicles release neurotransmitter, that membrane is recycled through this process and then reloaded with neurotransmitter. But that's a different story, but it is all the same idea of endocytosis. So endocytosis here. Yes?
What happens after the bacteria gets digested?
So what happens after the bacteria get digested? Then basically we're going to have a bunch of peptides and amino acids in here. And these will be broken completely down into their own selves. And then they can basically, we'll see in a moment, that early predatory cells would use that as a source of nutrition. We're also going to, when we get to the nervous system, when we get to the immune system, we're going to see that bits and pieces of these proteins are going to be displayed on the surface of these macrophages. But let's not go that far down that road just yet. We'll come to it later. So there are many fates of these proteins. In many cases, they're just broken up and discarded. But in other cases, they're broken into larger pieces and sent out to the surface for part of the immune system to recognize. Yes?
Would this only happen in like multicellular things to get rid of bacteria, or would it happen in single cellular things?
Will this happen in single cellular organisms? It probably will, but in single cellular organisms, these can be more or less predatory. So they're out looking for something to eat. And that segues nicely into what I want to talk about next. And what I want to talk about next is the idea of the mitochondrion. So we're going to take another cell here. And in all of our cells, we'll see the structures in the electron microscope. They look a little like this. They have this interesting crenellated internal membrane. But what characterizes them in part is they are double membrane. these structures, the mitochondria, these guys function to use oxygen as the final electron acceptor to generate ATP, adenosine triphosphate. We'll see later that ATP is the chemical energy that we're going to use for movement. But the lion's share of the ATP inside cells comes from this process of using oxygen, accepting using the oxygen as the final electron acceptor, and then making ATP from that. If you take a biochemistry course or a cell biology course, you'll follow through all of the intricate mechanisms for moving electrons across this membrane, moving hydrogens across the membrane to generate ATP. We're not going to get into that here. Just know that we make ATP here. Now, scientifically, I think this structure, this organelle, has an interesting history. And if we go back just a few years, the mid-1950s or so, and when it was the early days of electron microscopy, and suddenly people could see aspects inside the cell they could never see before. And there was a scientist named Lynn Margolis. And Lynn Margolis looked inside the cell. She was an electron microscopist, and she was looking at these structures, and she said, those are not like the other organelles. There's something weird about them. And she said, well, for one thing, they have two membranes. And having been a well-trained biologist, she said, well, those double membranes remind me of something. They remind me of bacteria. So bacteria often have double membranes. And so she generated a hypothesis. She generated the hypothesis that these mitochondria were captured a long, long time ago by some cell, and that the mitochondria are basically separate creatures, and that they're more closely related to bacteria than they are to eukaryotic organisms. Well, it's a great hypothesis, but you have to prove it. You have to test it. So what she did is she grew up plate upon plate upon plate of cells, scraped the cells off the plates, broke the cells up, and isolated the mitochondria. And she asked a simple question. Is the DNA and the RNA, are the nucleic acids in the mitochondria the same as what we see in the cell nucleus? It didn't take long before she recognized that the DNA and RNA, the nucleic acids in the mitochondrion, were a different sort from what we see in the nucleus. They weren't like chromosomal DNA. And in fact, they were very close to they're very close to prokaryotic DNA and RNA. They're much like bacteria. So that generated a hypothesis that's widely accepted now. And this goes back to one of the questions that came earlier. I think what happened is sometime a long, long time ago, probably 1.5 billion years ago or more, that there was a capture event. I should say, too, as part of this story, is that for most of the early history of the Earth, for the first couple billion years, at least 1.5 billion, the atmosphere was what they call a reducing atmosphere. There wasn't any oxygen. All of the oxygen we have in the air around us comes from photosynthesis. It comes from the result of plants or individual, in this case, the early story was cyanobacteria, absorbing a photon of light, using that photon of light then to split water. Splits water into hydrogen ion and an oxygen, and the oxygen then fuses with another to make O2. So the O2 in the world around us comes from photosynthesis. For much of the world's history, there wasn't much in the way of oxygen. Then the cyanobacteria come along, they make this oxygen, we get an oxygen-rich environment, but that's poisonous to many, many cells. Almost all cells don't like the oxygen. But it looks like some cells evolved a means of using the oxygen, and some cells, some bacteria used a means of using the oxygen to make ATP. And what we think is one of those bacteria was captured, much like this, by an early predatory eukaryotic organism. It was captured, brought into the cell, but something went wrong. It escaped from the vesicle, got out into the cytoplasm. And now this cell using the bacterium as a symbiotic organism to make ATP. Gave that cell a tremendous selective advantage. We think that all the life we see on the planet around us today, almost all of it, is related to that single cell. Because both plants and animals contain mitochondria. And if you look at their mitochondria, they're similar. They have similar DNA and RNA structure. So there was probably an early capture event that gave cells mitochondria and allowed them to use oxygen to make ATP. Then there was a capture event, and those of you who are botanists will know about this. Somewhat later, there was a capture event where there was a cyanobacterium that was captured in the cell. And then by capturing the cyanobacterium, those cells, the lineage of those cells now, could use sunlight to make carbohydrate. And so they became photosynthetic. But for us, we're not on that lineage. We're on the lineage that captured the mitochondria, but not the photosynthetic bacterium. And if you look at the details of the structure, much of the structure here looks much, much like the old bacteria that used to use flagella to run around. And those flagella seem to be related to the ability to make ATP. If you take a cell biology course, you'll learn all about it. It's a great story. But for now, just know that we think we've captured these structures and that they have their own DNA and own RNA, so their own nucleic acids. There's another peculiar characteristic of the mitochondria that's important for human history and studying human history. And that is, if we start out, of course, we all start out this way, as the ovum. The ovum, of course, is the egg. This is produced by your mother. And then along came your father's sperm. And your father's sperm was carrying half of his genetic material. And your mother's ovum was carrying half of her mother's genetic material. Now, that ovum contains mitochondria. So there are mitochondria in this thing. And it turns out, if we look closely at the sperm head carrying the nuclear material, then it has this tail. One tail, we've only got one tail in there. It has one tail, and around the one tail is a mitochondrion. And the force of the flagellum is generated by using ATP produced by this mitochondrion. But the weird thing is when this head of the sperm fuses with the plasma membrane of the ovum and delivers its nuclear material, the mitochondrion does not become incorporated into the ovum. What this means is that in all eukaryotic, well, at least in all mammals, I think at all chordates, then the mitochondrion is going to be inherited from the mother. So all of your mitochondria come from your mother. All of those came from her mother, which came from her mother, all the way back to the beginning of humans. And we've been able to use this to trace the movements of humans on the planet. Because there's a regular rate, more or less regular rate, at which there are mutations in the DNA of the mitochondria. And if we go back to Central Africa, we look in Central Africa where humans came from, we find that's where we get the greatest variability in the genetic material of the mitochondria. But as people fan out from Central Africa, they become smaller and smaller communities. The communities become isolated. And so when you begin to look at these smaller, later communities, then the variability in the mitochondrial DNA gets less and less. And by using mutations in the mitochondrial DNA, we can follow the migrations of many of the people on the planet and follow their matrilineal lineages. So it's been very useful in talking about human evolution. It's been very useful in understanding migrations. And it's been useful forensically in tracking people's maternal lineages. Yes?
Going back to that first cell, when the first cell like incorporated in the mitochondria, how does the mitochondria know to replicate with that cell?
It's a great question. And there have been arguments about this for a long time. How are the mitochondrial replications in fact regulated? And my understanding is it's random. It's just you have so many mitochondria in each cell that almost always they get divided up more or less evenly, and somebody gets a few mitochondria. And then they can divide and make more. But they do divide and make more and make copies of themselves. But it looks like it's just a random distribution. Yes?
I apologize if you mentioned this earlier, but is there a reason that the mitochondria from the sperm is left behind, or is it just...
Oh, the question is there a reason? That's always a hard one to answer. I think that the best reason I can come up with is that it doesn't matter. That is, the purpose of the sperm is to deliver its load of DNA to the ovum so that we then can combine the father's and the mother's DNA to make the offspring's full complement. It doesn't, I mean, it's used up its mitochondria, so there's no reason for it to go in. I have read places where there are rare cases where you'll see paternal mitochondria in organisms, but it's really, really rare. So I think that it didn't matter. It wasn't necessary, and it's probably something to do with the fusion event, which doesn't allow this mitochondrion to get into the cell, and you're only moving the DNA, the nuclear DNA from the sperm into the egg. I think that's the best answer I can give you. Let me take the one back there, and then we'll come around.
This is kind of changing gears just a little bit, but do they think that the mitochondria had that double membrane when it was first introduced to the cell or not? Because that's a very interesting thing. Most other organelles don't seem to have that double membrane.
So the question is, when do we think this double membrane occurred? My understanding is they think that the double membrane was there originally. Now, if you push me on that, I'm going to say I don't really know. Because, and the reason I'll say it's confusing to me is that when you look at a double-walled bacterium, there are lots of them, you will see that the flagella are all arranged on the outer membrane. They're not on the inner membrane. But yet, when we look at the ATPases, which are making ATP, which are very closely related to those flagellar rotors, those are on the inner membrane. So your question is well taken, and I don't have a good response to it. Someone knows, I just don't know. But why are the ATPases in the inner membrane? And as far as I know, they're on the outer membrane of bacteria. It's because they have to be in the outer membrane to drive the bacterium. And by the way, if you get into this, the flagella that we have here in sperm are different than the old flagella on bacteria or that used to be on the mitochondrion. But whether there was something that moved them, I don't know. But it's certainly true. Much of the understanding of how the mitochondrial inner membrane works was derived from an understanding of how the flagellar rotors worked in bacteria. And both in cases, they're using ATP and hydrogen ion gradients. There's another question. Yes?
What are the consequences of the cases that do have paternal mitochondria in those rare cases? Are there like implications to the rest of the human body or not really?
So what happens when there are paternal mitochondria? I think these people are living to adulthood. And then you have questions of what happens in the next generation. I don't know if those are all males or all females. I don't know really much more about it. But as far as I know, there are no deleterious effects. But maybe that was in one individual. Maybe that's the individual survived. I mean, there could be any number of explanations.
I just wasn't sure since it was a rare case.
It's really rare. It's really rare. But they have found a few of them. And it confuses things. Yes?
So macrophages the main type of cells, or are there other ones you have to differentiate between?
Say it again?
Are macrophages the main types of cells that we're focused on, or are there other ones that we have to differentiate?
So a macrophage is a very specialized kind of cell. The macrophage is a specialized cell that's part of the immune system. And you have them in your body now. They're out and about scouting for foreign invaders. And they'll bind things in this way, bring them in, and destroy them. But that's a specialized form of cell. But I use that specialized form of cell as an exaggeration of how endocytosis works. And I don't want to leave you with the idea that other cells don't have endocytic mechanisms. They all do. And they use those endocytic mechanisms for a variety of things. Anything else? Yes?
Do the mitochondria have their own means of reproduction, or do they
Do the mitochondria have their own means of reproduction? They seem to be able to reproduce on their own, as far as I know. But I have to be really careful of that, because although I glibly tell you that the mitochondria have their own nucleic acids, both DNA and RNA, those genes are almost exclusively for ribosomal RNA and transfer RNAs. And if you study mitochondria, you find that the mitochondria require a whole set of proteins that are generated by the genomic DNA. So the mitochondria are not just these bacteria living inside our cells independently. They require a whole host of proteins to be brought into them, and those proteins are proper proteins made from genes in the nucleus. And there are many of them. I think someone told me like there are 900. It's a big number. So you're right in the sense that, yes, these are symbionts, but they're not autonomous. They can't survive on their own. And many of their functions require that we use genes from the nucleus. Anything else? Yeah?
Is their dependence on survival on the cell new, or is that something that they had to do previously in their bacterial form?
So are you asking about the mitochondria or the cell that captured it? The mitochondria, we think the mitochondria used to, a long, long time ago, this is like 1.5 billion years ago, maybe more. These guys were out and about, and they lived on their own. And then they were captured. And then they've been modified. They've themselves have evolved over the last 1.5 billion years to become proper mitochondria. And no longer can we turn them loose and have them live on their own. Does that answer your question? Okay. Well, no more questions. Let's kind of, yeah. The next thing I want to talk about is the cytoskeleton. You may remember that the cytoskeleton is a characteristic of eukaryotic organisms. And there are 3 main cytoskeletal proteins. The first ones are called microtubules. The second one is called filamentous actin. So filamentous actin. And the third one is intermediate filament protein. Now these first 2, these are the ones I'm going to, well, let's do all 3 of them at one time. What all of these have in common is that they are polymers of smaller proteins. Microtubules, these guys are polymers of tubulin. Filamentous actin is a polymer of globular actin. And often you'll see this referred to as G-actin. And you'll see the filamentous form referred to as F-actin. Similarly, intermediate filaments. And intermediate filaments come in 2 big varieties. One is keratin, and the other is neurofilament protein. And these 2 are polymers of either keratin monomer or neurofilament monomer. The intermediate filaments seem to be primarily structural. They don't do much of anything else. They're basically like stuffing inside the cell. But the microtubules and the filamentous actin are the ones I want to really emphasize, because it's the microtubules and the filamentous actin that support vesicular transport. And I can even say here they support vesicular transport using molecular motors. But to understand how those motors work and to understand how this ties together, we first have to spend a little bit of time forming those polymers. So I'm going to take this panel and I'm going to imagine how we can form these polymers. Now this is going to be nothing like how it works in biology, but we'll get the idea of how we'll take monomeric proteins of tubulin, a dimer, and stick them end to end and side to side to make these structures. The actin will form end to end, but it won't form much side to side, so it doesn't make a tubule, it makes a filament. So let's start here with the discussion of microtubules as polymers of tubulin. So we can talk about microtubules as polymers of tubulin. Now, tubulin is this sort of peanut-shaped molecule. Looks a little like this. Itself, it's a dimer. It has an alpha subunit and a beta subunit. But these peanuts can link end to end. So this alpha subunit can link to its neighbor, alpha, beta. These are meant to be identical. Of course, when I draw them, they're not. Alpha, beta. Alpha, beta. So they're stuck end to end. They're also going to be assembling side to side so that we can look at them this way. Alpha, beta, alpha, beta, alpha, beta, alpha, beta. Now, if we would continue this on to its logical conclusion, take these alpha, beta subunits, the tubulins, assemble them side to side, we get a great sheet of those. We don't get that in biology. We can make them in the laboratory, but we don't get that in biology. Turns out that when these guys assemble side to side in biology, there's a slight curvature from one tubulin to the next. And if we look at these end on, if we take this great long string and cut it, what we see is a typical arrangement of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. There are going to be 13 tubulins in a circle. This tubular structure is about 24 to 25 nanometers in diameter. But it's exquisitely long. It's extraordinarily long. So if you imagine this microtubule extending behind the plane of the whiteboard and out into the audience, this would go for, at this dimension, tens or hundreds and hundreds of meters. So it's a long, thin molecule. And it's a long, thin molecule that's produced by polymerizing tubulin. There's another important characteristic I need to point out here, in that these molecules have endedness. And what I mean by endedness is it's the way they assemble. There's an end here to which we add tubulin. And there's an end here where tubulin leaves. This gives a kind of endedness to the molecule, but it also gives us a way to describe the molecule itself. It tells us that where we add on is the plus end, and where the tubulins come off is a minus end. So now we have a plus end and a minus end to this microtubule. Now it's true in biology, there are times in biology where we'll add tubes to one end and they fall off, and they're what we call treadmilling. They're constantly moving around. We put them on, we take them off, it continues. But that's not the case of what I'm going to draw now. What I'm going to draw now is the case of a neuron. And I want to use a neuron because it's an asymmetric cell. And these asymmetric cells, we see that there's what is known as a neuronal cell body. It looks a lot like what we've just talked about for a garden variety cell. There's the nucleus, so we'll have endoplasmic reticulum. But then coming out of that cell body is this great long process. In some cells in our bodies, this process can be, a meter or so long. There are motor neurons in your spinal cord which will extend out into your hand, and those will be close to a meter long. Even longer in something like a giraffe. So they have these great long asymmetric structures called axons. The cell body here is the site of protein synthesis. So this cell has a logistic problem. It has a problem in that all of its proteins are going to be made here, but they may be used throughout the cytoplasm along the membrane on this great long structure. So how does the cell solve this logistical problem? Proteins in one place and using them in another. The first step in that discussion has to do with the microtubules. They're here in the cytoplasm. Now, these are long. These may be several hundred micrometers long, and they're very narrow. But when they began to look at microtubules in the cytoplasm of neurons, they found they were always oriented in one way. They were always oriented such that the plus ends were farther away from the nucleus, and the minus ends were closer to the nucleus. So this consistent orientation said there must be some reason why we orient those microtubules in that way. And shortly after that, they found that vesicles could be moved along the microtubules. And let me spell it out here and say that microtubules are the substrate for vesicular traffic. The first character we're going to talk about is something known as kinesin. Kinesin is said to be a plus end directed motor. Oh man. So here in the back board, let me draw a kinesin molecule. I can think about it a little bit. A kinesin molecule looks a little like this. It has two alpha helical regions. And at the bottom it has a kind of foot. These two alpha helical regions that are intertwined, a kind of a foot. At this end, the kinesin are what is sometimes known as light chains or adaptin proteins. What they do is they bind to receptors on vesicles. So there'll be receptors, transmembrane proteins in a vesicle, and those will bind to the adaptins. And this now attaches the vesicle to this end of the kinesin molecule. This end of the kinesin molecule binds to the tubulins. So the feet bind to the tubulin. And the feet then walk along the microtubule. They walk in 8 nanometer steps. It turns out that these tubulins are separated by 8 nanometers. So where I leave you is this idea that these kinesin molecules are going to walk from one tubulin to the next along the microtubules as though the tubulins were stepping stones, and they're going to walk in one direction toward the plus end. And tomorrow we'll finish the story, or Monday we'll finish the story, talking about how these are ATPases. And each time they cleave a molecule of ATP to ADP, they walk, the one leg flips over the next, walking along the microtubule. And with that, I'll let you go.